So we're interested in structural functional relationships in protein, and specifically light-activated proteins, which undergo large conformational changes in response to light absorption. So in this experiment, we're interested in a light harvesting antenna complex from Cyanobacteria. And these antenna complexes are extremely important for regulating how much photon flux reaches the photosynthetic reaction centers. So in this experiment, we aim to measure, extract structure from phycobilisomes by doing single particle imaging. And previous single particle imaging experiments were done mostly in aerosols because you get a large background due to the solvent. In this case, water or some aqueous buffer. So in order to do the experiment in solution, which we aim for this week, we needed to develop a different type of liquid sample injection, which is the form of a very thin liquid jet on the order of sub 50 nanometers, which reduces this background by upwards of a factor of 50. So the instrument here at SPB is specifically conducive for using these liquid jets, unlike other instruments which have very small vacuum chambers. SPB has a large vacuum chamber which is able to handle the large liquid flow that are characteristic of these types of liquid jets. That, together with a high repetition rate and both of the X-rays and the detector, makes it uniquely suited to carry out this type of single particle imaging. So recently there was a cryo-EM study on this same phycobilisome light harvesting antenna, and they found that the structure was what was originally thought to be one single structure was actually a mixture of three separate conformations. So phycobilisome is composed of a central core and outer rod sections, and the rods were found to exist in either an up conformation or a down conformation, and it's suspected that the position of these rods has directly modulates the funneling of the energy into the photosynthetic reaction centers. And since in most photosynthetic systems they often have too much light, it's very important to understand the position of these rods and the relationship with how these organisms deal with situations of under high light exposure, which can lead to degradative effects.